This is the video for Lab 11, Carbohydrate Metabolism. When identifying unknown bacteria, one of the first things that is done is a gram stain. Doing a gram stain and looking at it under the microscope will give an individual two important pieces of information. The first piece of information is the shape of the bacteria. And there are basically three shapes to choose from: cocci, bacilli, and spiral. The other piece of information that you can get from a gram stain is the gram reaction. This is determining if the cell wall is a gram-positive cell wall or a gram-negative cell wall. So after doing a gram stain, you would be able to tell if your unknown is a gram-positive coccus or a gram-negative coccus. Gram-positive bacillus, gram-negative bacillus, gram-positive spiral, gram-negative spiral. And that is important because it allows you to eliminate all the other options. But just knowing the shape and gram reaction will not give you a positive identification of your bacteria. In order to identify the exact genus and species name of your unknown bacteria, you need to do a biochemical profile. Metabolism is defined as the sum of all the biochemical reactions in an organism. And just as an example of some of the reactions in an organism, this is a map of several different types. Carbohydrate metabolism is here in the middle, then other metabolic pathways like lipid metabolism, nucleotide metabolism, amino acid metabolism, and so on. Not all bacteria can facilitate all reactions. So a biochemical profile is getting a list of the enzymes or the reactions that the bacteria is capable of performing. And to do this, we can't see the actual molecules or reactions taking place, so we use indicators. Today we are looking at carbohydrate metabolism, so that small segment of all the metabolic reactions in the cell. And carbohydrates, remember, are used to generate energy. So that is the purpose of carbohydrate metabolism. If a bacterium does not have carbohydrates to break down to produce energy, they can use an alternate pathway from fat or using proteins. The first test that you would be doing would be using the Phenol Red Broths. So the indicator in this experiment is phenol red, and phenol red is red around neutral, and then as the pH drops, it becomes yellow. As pH increases, it becomes pink. And the pathway you are looking at are the different sugars like sucrose and glucose and some other sugars. So you are determining if the bacteria has the ability to break these sugars down to generate energy. And if they do, they will produce acidic byproducts. So if the bacteria can utilize the type of sugar you're testing for, then the pH should decrease as the acidic byproducts build up in the test tubes. Some of the results you could see, these are glucose tubes. Glucose tubes also have a Durham tube inside. The Durham tube is used to trap carbon dioxide that may or may not be produced when the bacteria ferments the glucose. So for these results, the media turned yellow, so it's positive for acid production, and there is a bubble in the Durham tube, so it is positive for gas production. This tube turned yellow, so it is positive for the sugar production or sugar metabolism, and there is no bubble in the tube, so it is negative for gas. This tube remained red, so it is negative for acid production. For this lab, you will be testing glucose with Durham tubes, then lactose, sucrose, and mannitol. And they all respond the same way. If the media turns yellow, it is a positive. If it remains red, it is negative for being able to utilize that sugar. The next test would have been starch hydrolysis. For this, you are looking at the bacteria to see if they have the ability to take starch and to perform a hydrolytic reaction to break off glucose off of that polysaccharide, which would go into glycolysis, Krebs cycle, electron transport chain to generate energy. For starch hydrolysis, you would have inoculated bacteria in a straight line on a plate, and after growing for 48 hours, you would have added iodine. When iodine interacts with the starch, it turns this black color. If the bacteria were able to secrete an enzyme to break down the starch, then the starch surrounding the bacteria would have been broken down into glucose, and you would get this halo around the bacterial growth where it turns just that light brown. So this is a positive. You see the bacterial growth in the center, and then the halo next to it. This is a negative because you have the bacterial growth, and it's a nice even black color. The next test is the Methyl Red-Voges Proskauer test, or the MRVP test. And for this, it's methyl red is the indicator. So methyl red has different colors at different pHs compared to phenol red. For methyl red, it is red at an acidic pH, and it is yellow when it is neutral or basic. So this has a different color profile than the phenol red. For methyl red and Voges Proskauer, you are determining if the bacterium takes the glucose and either goes down an acidic pathway or goes down a neutral pathway with how it breaks it down. So the MR test is determining if the glucose is converted to acidic byproducts. The VP test is determining if the glucose is converted into neutral products. And for each of these, a red color indicates a positive for those tests. This is a methyl red test, the methyl red results. After you add the reagents, if it remains yellow, it is negative. If it turns red, that is a positive result. For the VP test, this is the VP test. If it is yellow, it is a negative. If it turns red, it is a positive. And for organisms, they cannot be positive for both methyl red and VP because these two pathways, it's an either or. The byproducts are either going to be acidic or they are going to be neutral. So a solution cannot be acidic and neutral at the same time. The last test is citrate utilization, and for this, the pH indicator is bromthiamyl blue. So bromthiamyl blue is a greenish color at neutral and a blue color when it's basic. And for the citrate utilization, you're looking at the source of carbon for metabolism. Most of the time, the bacteria are using carbohydrates as their source for carbon, but they can also use citrate if carbohydrates are not present. So if the bacteria are able to utilize citrate to provide carbon for their metabolism, then the media will turn blue. If they are unable to use citrate as a source of the carbon for metabolism, the media will remain green and the bacteria are unable to grow. Once you have started to build your enzyme profile or biochemical profile for an unknown, then you would take that information and compare that to known profiles. When you have acquired the data for your unknown bacteria for all the different experiments and have built an enzyme profile or biochemical profile for your unknown, then you would go to the dot matrix chart for determination of the identity of an unknown microorganism. This is uploaded onto Eagle, and you would compare your unknown's profile with known profiles. And I'm sorry here, but this has the names of the knowns, and in this copy, for some reason, they were cut off. But I just want to tell you in this matrix that, of course, a positive is a positive result, and a negative indicates negative for those results. I do want to mention that D is determinative. That means it's variable. And blanks are tests that are not traditionally done for those types of bacteria, and that's also mainly because they have variable results. So once you get some information from the carbohydrate metabolism of your unknown, then you can start comparing your unknown to known profiles.